All right, so what we're going to do today is we're going to go over what we call complex inheritance patterns. So at this point, you should have done some practice with basic Punnett squares. You should be able to look at it and know where the parent genotypes go. You should be able to look at the offspring, tell me what the phenotypes are or what they look like, what the percentages and everything like that are. If you are not to that point and you still need to practice, let me know, email me or something like that, and let's get you guys figured out with this, because you're going to have a basic understanding of Punnett squares and how they work before we can get into our complex inheritance patterns that we're going to talk about today. If you called in to Zoom into class, great. If you need a little bit more help one-on-one, I would recommend watching this video. And those of you at home, oh my gosh, it's the end of the day and I'm tired. I'm going to work you through the notes for today, a couple of the practice things, and then there is a worksheet that has a few practice Punnett squares on it that I want you to do for what's called incomplete and codominance. So I'm going to go ahead and pull up the notes page. If you guys would open up your digital notebooks, we're on page 25 today, and we're going to do page 25, 26, 27, 28, 29, 30. No, we're not going to do 30. I want you to delete page 30. I want you to delete page, so 30, 31, so 30 and 31. Delete those two pages. Okay. So basically we're going to hit page 25 to the end of the notebook today. Nope. We're going to delete. We're going to delete these ones. So 32 through 34, delete those two. All right. We're not going to have time to get into those before the end of the quarter. So what we're going to look at is in heterozygous, well, I guess in what we've been looking at lately is what's called Mendelian genetics, or where everything's determined by two alleles. There's either a dominant or a recessive. There's nothing in between. So if an organism inherits one or two copies of the dominant allele, that trait's going to show up. If they inherit two copies of the recessive allele, then that trait is going to show up. If they have a dominant with a recessive, the dominant trait is going to show up. So we've had one or the other. It's either dominant or recessive. What we're going to get into today is what happens if it's not quite that easy. So what happens, what we've been saying is that, like, if you have a red flower and we cross it with the white flower, red would be dominant, white would be recessive, and red would always trump over white. What we're going to look at today is what happens if I breed a red flower to a white flower and it comes out red and white striped, or I mix a red flower with a white flower and it comes out pink. What happens with that? So if you would please follow along in your notes, I'm going to go to the PowerPoint now. I should be able to pop up the little words that you need to drag and drop into place with this. So here we go. So when we talk about these more complex patterns of inheritance, we're going to look at just the heterozygous, right? So normally we've been looking at a dominant trait. Homozygous would be dominant, dominant. Homozygous recessive would be recessive, recessive. We're going to focus on the heterozygous, a dominant and a recessive. And that's going to be our focus for the next couple of days, is what happens with those heterozygous genotypes, big letter, little letter, and how does that affect how certain traits are inherited. So in most of the time, in a heterozygous offspring, the visible phenotype isn't always the dominant or the recessive trait, right? Most of the time when we have what's called Mendelian genetics, we crossed out Gregor Mendel here because he's the one that did the whole peas, tall peas over short peas, green peas over yellow peas, purple flowers over white flowers. Most genetics don't necessarily work that way. Most genetics are more complex. So we're going to be referring to these as what's called non-Mendelian genetics because they don't follow the pattern that Mendel set together. 90% of the time or so, most inheritance that you're going to see is not going to be, it's either dominant or recessive, okay? When we did the human traits, we looked at like the widow's peak in the hairline, or being able to roll your tongue, or having a hitchhiker's thumb. Not even those are strictly one or the other. Okay, you can have people with a really well-defined widow's peak You can have people with kind of a middle widow's peak, and you can have people with no widow's peak. Those are the ones that we had. Hitchhiker's thumb, you can have people whose thumbs bend completely backwards, people whose thumbs are kind of in the middle, and people whose thumbs just go straight up and down. So even with those traits that we looked at, it's not one or the other. There's a range in the middle of it, okay? So it's not always dominant or recessive. Sometimes it's a combination of traits. We combine a little bit of the dominant with a little bit of the recessive, and we get something new, all right? So the first one we're going to look at is what we've been talking about, is complete dominance. So in this instance, the blue monster would be dominant over the yellow monster. So if I breed a blue monster to a yellow monster, I get a blue monster, okay? The yellow is recessive. It doesn't get to show up. The only way I get a yellow one is if maybe this blue guy's heterozygous and he has a yellow in there, okay? Our first one we're going to look at is something called codominance. And what happens in codominance is both alleles are going to be dominant alleles, and they like to show up together, all right? So for our example here, we have a white cow and a brown cow. Now, the white is dominant, and the brown is dominant. So when I cross a white cow to a brown cow, I don't get a brown cow or a white cow. I get something in the middle. So I get what's called a roan cow, and that's basically a white cow with red spots, okay? So in codominance, both of the alleles are dominant. So both traits are going to be seen equally. Maybe one's a little bit more than the other, but you will see both traits. So on this one, we have our white cow here, and we have our brown cow, and then all of the offspring have a combination of both white and brown. So codominance, something, if you have a coworker, it's somebody that you work together with. So a codominance are going to be two traits that essentially work together, or they both show up. Your coworkers show up to work with you. Codominance, both traits want to show up. So for example, if we cross a white flower with a red flower, I wouldn't get just either a white or red flower. I may get a flower that shows both of those traits in it. Okay, so like our picture here, we can see the white around the edges. We can see the pink, the red in the middle, all right? It's not one or the other. It's a combination of the two. And because it's codominant, we're going to see both traits, right? So that's the one thing I want you guys to remember, this codominant, both traits show up. So if we look at our monsters, we have a blue monster that we breed to a yellow monster, and we get a yellow monster that has, or a blue monster with yellow polka dots. We have both traits showing up here. Or if we want to look at it from the bear point of view, we all know this is not how panda bears are formed, but if we take a white bear and we breed it to a black bear, we get a combination of both black and white bear. So both the black traits and the white traits are showing up together with that. So if we look at this Punnett square that we have here up on the screen, okay, we're going to say there are two alleles. We have, for a hair color, we're going to go really outrageous with our hair color here. We have a red trait and a blue trait. So a red allele, we're going to say is R, and a blue allele is B. So what would be the resulting phenotype of a homozygous red, meaning it has two red alleles, and a homozygous blue, which is two blues, if the alleles show codominance? So I'm going to come in here, and this works the same way as normal Punnett squares do, except we're using different letters. So when we did inheritance before, we used the same letter. So like for hair color, we'd use an H for dominant and a lowercase h for recessive. With codominance and incomplete dominance, we're going to use a separate letter for each allele. So we have R for red and B for blue. So I'm going to come in here, I'm going to take my R's and put them in my Punnett square. Those are bad R's. And then I'm going to bring my B's across. And fill in that Punnett square. All right. So I have individuals that have both a red allele and a blue allele. Well, since they're codominant, they're both going to show up. So my options are they all red? Are they all blue? Are they purple? Or would they have red and blue patches? Well, since it's codominant and both alleles get to show up, it's going to have red and blue patches. So maybe you have little streaks of red and streaks of blue all over in your hair. All right, that would be an example of codominance. Two dominant traits that both want to show up and they both get expressed equally. Why can I not get? Okay, so let's go ahead and look at the Punnett squares in your notes for codominance. And I'm going to close this out just to make sure that I get it closed out. Okay, I'm going to stop my share. I'm going to go back in because last time I did this, it didn't transfer well. So now we're going to go into your notes. And right now your note should look like this. In heterozygous offspring, the visible phenotype isn't always the dominant or the recessive trait. Sometimes the phenotype is a combination of traits. In codominance, both of the alleles are dominant. So both traits are seen in the phenotype. So example, a white flower is crossed with a red flower and their offspring are, what did I put on this? Should be blended. So you both. I'll have to look at that one. I don't know what I put on there. All right. So let's look at your Punnett square for this. So we're on slide number 27 right now if you've deleted everything. So our first question here says, when red cattle are bred with white cattle, they produce roan, or red and white hares, offspring. If two roan cattle are crossed, what is the probability of red, white, and roan colors in their offspring? So if a cow is red, okay, it's going to have big R, all right? R means red. If it's white, it's going to have WW. W stands for white. And if it's roan, it's going to have one of each. It's going to have a red and a white. So in my cross here, I'm crossing two roan. So I have a red and a white on top, red and a white on the bottom. So we're going to go through and fill out our Punnett square. So I wrote my R's down, W's down, R's across, W's across. So I have to look at the percentage of these. So I think these are a little bit easier than traditional Punnett squares because we're not worried about the combination of different sizes of letters. We can just look at the combination of letters. So RR means red. Well, I only have one right here. So that means that I have 25% would be red. White, there's just this one here that has two W's in it. So that's 25%. And then roan has both a red and a white. So I've got one here and one here. So that leaves me with 50% roan. Our second one says that in dogs, gum coloration is codominant. So you guys ever looked at a dog's gums? Some of them have pink, some of them have black, some of them have kind of spotted gums in there. It kind of creeps me out. But the dog's gums can be black, pink, or pink with black spots. What is the probability of having puppies with pink and black spots if you cross a dog with pink gums? So we're going to say that is PP, okay, pink, with a dog with pink and black gums. So that's going to be PB, so for pink and black, right? So my pink ones are going to go up here. I'm going to color this in pink so I know what color it is, okay? And then my pink and black is going to go here. So I'm going to color this one in pink. And I'm going to color this one in, I'm going to do it gray because I don't want it to, maybe I'll make it black. And I'll just make my text white. Okay. So what's going to happen now is we're going to do our cross just like we would. So I'm going to bring my P's down. So P, P, and P's down here. P, 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 down here. I'm going to bring the P's across, or the P across. So P, P, and bring the B across. So B, B. All right. So what percentage of my offspring here are pink, meaning they have two pink alleles? Well, there's one here, and there's one here. So these are both going to be pink. So that's 50%. And then these ones down here are pink and black. So do I have any that are just black? I'd have to have BB right here, which I don't, so it's 0%. And then what percent are PB or pink and black spotted? Well, that's these two right here. So that's 50%. So that is codominance. Now let's go back into our PowerPoint. I'm gonna stop my share again. I'm gonna restart it, so hopefully I'm doing this right. All right, and we're gonna pick up with incomplete dominance, which is gonna be right here. All right, so in incomplete dominance, we don't have a dominant or a recessive gene or allele like we did before, but instead of both showing up like they do in codominance, incomplete dominance is going to blend them together. So a couple times when we were talking about the dragons today, somebody asked me if they could do orange eyes because they had a red allele and a yellow allele. So red plus yellow equals orange. Well, what we were doing today was complete dominance. So you had either red or yellow. Incomplete dominance, what we're looking at right here, you could have had an orange eyes or orange eyes, but we weren't there yet. So when we're talking about incomplete dominance, we're talking about both alleles are blending together, so you can't see where one starts and one ends. All right, so the alleles are neither dominant or recessive. They mix together in heterozygous individuals. So our picture here is we have a red flower with RR, and we have a white flower with WW. And if we mix them together, we get pink flowers that have RW as their genotype. So again, we would have two dominant alleles here. We'd have two dominant alleles here, but when they are inherited together in the heterozygous genotype, they're expressed as pink. They're expressed as an equal blend of the two. Okay. So same thing here, a white flower is crossed with a red flower and their offspring are pink. You see a blend of the white and the red. So it's like pouring your red flower in and pouring your white flower in and shake it all up and you get pink flowers. Best thing of it, like mixing paint is the best way for me to explain incomplete dominance, is you take one and the other, shake it together in a mixer, and you get the mess. Okay. So for example, with our monsters like we did before, with complete dominance, we bred a blue to a yellow and got yellow. In codominance, we bred a blue to a yellow and got blue and yellow. In complete dominance, I breed a blue to a yellow and I get green because blue and yellow makes green. Or if we want to look at it this way, duck plus beaver equals platypus. They're blended together. So in our previous one, we saw white bear plus black bear equals white and black bear. Okay. If we're looking at the bears again, we would see white bear plus black bear equals gray bear in incomplete dominance. So codominance, you see both traits. I would see black and white. Incomplete dominance, they're both blended together. So I would see white bear plus black bear equals gray bear. Okay. So once again, look at this Punnett square with hair colors, our crazy, crazy hair colors. We're going to use the exact same question. There are two alleles for hair color, red with an R, blue with a B. What would be the resulting phenotype of a homozygous red, so RR, and a homozygous blue, BB, if the allele showed incomplete dominance? So again, in here, we're just going to do like we did before. We're going to take the R's. It's so hard to draw R's with a mouse. And then I'm going to come in here and put in my B's. These are almost worse. Oh, they're so bad. Okay, so I have all of my genotypes now are RB. And since this is incomplete dominance, and I'm going to see a blend of the two traits, I'm not going to have red, okay, because there's this B in there. I'm not going to have blue since there's this R in there. But because it is incomplete dominance, which means that those two alleles are going to blend together. If I mix red and blue, what do I get? I get purple. Okay. And if you remember, red and blue patches was the answer that we had for codominance because we'd see both of those equally. Clear all my drawings. Okay. So back to your notes for Punnett squares. I'm stopping and starting. Okay, get this out of here. All right, back in your notes on page 28 now, we're going to do to practice incomplete dominance. So if you look on the side right here in the green, it says incomplete dominance. So that's where you know where we're at. In snapdragons, flower color is controlled by incomplete dominance. Two alleles are for red and white. So red would be R, white would be W. The heterozygous genotype has a pink phenotype. So if it's heterozygous, it means it has one of each. So that's going to be RW. If I cross two pink snapdragons, what are the possible outcomes of the offspring using percentages? So I'm going to cross two pinks, which means that each one has an R and a W, and an R and a W, plus red plus white equals pink. Nope, this one. So I'm going to fill in my Punnett squares. I brought my R's down, W's down, R's across, W's across. So what percentage of my flowers here would be red, meaning they have two red alleles? Well, it's this one right here. This one is red. My percentage would be 25%. White is this one right here, and there's only one that has two W's in it, so that's another 25%. Oops, that's the wrong one. 25%. And then RW, red plus white equals pink. So this one right here is pink, and this one right here is pink. So what's my percentage of pink flowers from this cross? Well, 50%. So if I breed two pink flowers together, I would expect that 25% of them would be red, 25% would be white, and 50% of them would be pink. So here, this one's kind of going to go with my white bear plus black bear thing. But in guinea pigs, there are two alleles for fur color, white and black. So we're going to say that white is W, goes without saying, and black is B. Okay. A guinea pig with a heterozygous genotype will have a gray phenotype. So if it's heterozygous, it's got one of each. So it's got a black and a white. What are the possible outcomes of crossing a black guinea pig with a gray guinea pig? So if he's black, he's going to have two B's, so black and black. And if he's gray, he's going to have one of each. So black plus white equals gray. So fill out the Punnett square. All right. So what percentage of these would be black? It's anything that has two B's in it. So that's this one right here and this one right here. So that gives me 50% are black. I don't have any white because I don't have any offspring in here with two W alleles or two white alleles. So that's 0%. And then down here I have one black and white and two black and white. So that gives me 50% of them that are going to be gray. So if I mix a black guinea pig with a gray guinea pig, I would expect 50% of them to be black, 50% of them to be gray. I can't get white unless some kind of random mutation comes in there. All right. Now, we've got one more thing to get through with this, and that is what's called multiple allele inheritance. And the only thing we're going to look at with multiple allele inheritance is human blood typing. All right. So blood typing is controlled by three alleles, three. There are 4 different blood types, though. You can have type A blood, you can have type B blood, you can have type AB blood, and you can have type O blood. All right. So when we're talking about multiple allele inheritance, we're going to say that some genes have multiple alleles, right? Which means that 3 or more possible alleles determine a trait, because most of the time it's 2 alleles. You have a dominant and a recessive, or dominant in the case of codominance and incomplete dominance. Most of the time we're going to look at 2, all right? Multiple alleles means that there's at least 3. Now, like eye color is controlled by about 7 different genes. Okay, so you could have 7 different alleles that control what color your eye is, and that's why you have such a huge range of eye color in humans. It's not just brown eyes, blue eyes, green eyes. There are people with gray eyes and people with violet eyes, people with light brown eyes, people with dark brown eyes, and bright blue eyes, and light blue eyes, and brownish gray eyes, and brownish green eyes, and there's a whole spectrum of it. Okay, so that's why we get such a range. Now, an organism can only inherit 2 alleles. So even though there's 3, 4, 5, 6, you only get 2 for each gene. Okay, still it's 1 from each parent. So you could get, out of those 7 genes for your eye color, you're still only going to get 2 for each gene. Okay, so 1 gene or 1 allele from each parent. Doesn't how many genes are coded for. You still only get 2 alleles for each one. So even if there are, nope, not 2, 3 or 4, not that. Come on. 3, 4, I don't want that. Work with me here. 3, 4 or more alleles, an individual can only have 2. That's all you get. Okay? So some genes have multiple alleles, which means that 3 or more possible alleles determine the trait. Remember, an organism can only inherit 2 alleles for each trait, 1 from each parent. Even if there are 3 or 4 or more alleles, you only get 2. All right? So in humans, blood type is controlled by 3 alleles. You have what's called the A allele, you have the B allele, and you have the O allele. Okay? So over here, we see that A and B are dominant to type O, which means that you have an A and an O together. The A is going to overpower it. But A and B are co-dominant with each other. So that's how I can have type AB blood. And type O is recessive. So that ties with the top one. So O is recessive to everything. A trumps O, B trumps O, but A and B are co-dominant, which means they can exist together. Okay? So for example, if you get an AA allele, you have type A blood. If you have an AO allele, you have type A blood. If you have BB, it's type B. If it's BO, it's B. If you have AB, it's type AB. And if you have OO, it's type O. Okay? So that little chart is right down here. Now, when it comes to blood typing, we don't use B for blood. We use something called, we use an I. So every time you're going to do blood typing from here on out, blood typing is always represented with an I. It stands for immunoglobulin, which is a chemical in blood. All right. And the way we represent it is type A is a capital I for dominant, and then it gets a little subscript A up there. So you can see that right in here. Okay. So type A is capital I with a little uppercase A. Type B is the same, except it has a little subscript A, or superscript, I guess. Type AB, you get 2 I's, IA, IB, and then type O, you get II. So technically type A, why can I not change this? That's why. So type A could technically be IA like that, or how do I do that? Or it can be IA little I. Okay. So this one right here would be what's called the homozygous version of it. This one right here would be the heterozygous version of it. Same thing with IB. So type B blood could be either IB for homozygous because they're both the same, or it can be IB, little I, okay? And then type AB is IAIB, type O is II because it's recessive, just a little bitty, okay? So what does all this mean? Oh, let's look at some Punnett squares, okay? So this one says, show the Punnett square and phenotypic ratio for the following cross. Both mother and father have type O blood. So I'm going to go back to my little chart here, and I see that type O is little I, little I. Well, since they both have type O, they each get little I, little I. So this is one of the world's easiest Punnett squares. Ta-da. It can only be little i's, that's it, okay? So your parents' genotype, you have little I times little I. The possible genotypes, well, there's only one genotype it can be. It could be little I. That's it. Possible phenotype, so what type of blood is this? So if it's little I, it's type O blood. Right? Now type O blood is nice because they can donate to everybody, but they can only receive blood from a type O. If you're type O negative, you're what's called a universal donor, which means you can donate to everybody, but you can only receive type O negative blood. That's it. Okay. Type AB is lucky because they can receive A blood, B blood, O blood. So they're the universal receiver. They can get from everybody. But type O can only receive type O blood. So phenotype percentages, well, it's type O and it's 100%. So if you have two parents that have type O blood, you have type O blood. Your siblings have type O blood. There's nothing else it can be. Now, let's look at one that's a little bit more difficult. So the father is type A homozygous and mom is type B homozygous. Remember, homo means same. So that means it's going to be this one right here, this IA. Okay, because they're both the same. So I'm going to come here, I'm going to go IA, and I'm going to make these, superscript. And then mom is IB, which means that the B is going to go superscript 2. And if you did a shortcut for this, it's just highlight it and then push Control and then the period button, and that'll bump it up there really quickly. So my Punnett square is going to look like this. I'm going to drop my A's down. So IA, drop these ones down, IA. Bring my B's across. So IB. I'm going to come up here and I'm going to fix these because the formatting is going to drive me nuts if I don't. And again, I'm just highlighting Control period. So the parents' genotypes, so dad was IA. Dad was, mom was IB. Nope, not that. Okay. The possible genotypes. So what do we have in our boxes right here that their offspring would have? Well, there's only one. It's IAIB. Which means that the phenotype So if we look back at our chart right here, IAIB means it's type AB blood. So that'd be the phenotype. So this is type AB. Phenotype percentages, type AB, and it's 100%. So if you have a parent who is type A homozygous, meaning that's they only have type A alleles in there, and a parent who's type B homozygous means they only have type B alleles in there, the only outcome can be type AB blood because AB is codominant. They both exist together. So that's why we have AB. It's A plus B equals AB. All right, let's look at another one. The father is type A heterozygous and mom is type B heterozygous. So if it's heterozygous, meaning you have one dominant, one recessive. So it's going to be this one here, the IA little I. So dad is type A heterozygous, so he gets an IA. This is his type A blood. And this, which means that he had a parent who had type O blood. That's where this one comes from, because O is recessive. Mom has type B heterozygous, which means that she also had a parent who had type O blood. That's where this one comes from. So now we're going to come in here. I'm going to bring my IAs down. Oh, stop it. Bring my I down. And bring my B across. So IB, IB. Bring my little I across down here. I. I'm going to fix my little B's here. All right. So parents' genotypes, we had IAI times IA or IBI. So they're heterozygous means they have a dominant, the A or the B, and they have a recessive, which is from a type O. Possible genotypes, well, we can have IAIB. We can have IAI. And we can have IBI. And we can also have II. So you have IAIB, IAI, IBI, and II. So possible phenotypes, well, this one right here is going to be highlighted. This is type AB. This one right here is type A. This one right here is type B. And then this one right here is type, oh, I'll shake those down just a smidge so they'll fit. All right. So the percentages of this, well, they're each only 25% because there's only one of each of these genotypes. So type AB is 25%. Type A is 25%. Type B is 25%. And type O is 25%. So technically, if you had parents with these genotypes, you could have siblings with every different genotype there is. So you could have sister with A blood and a brother with B blood and another brother with AB blood and a sister with O blood. Everybody can have a different blood type in your family. It's totally possible. So one more on here. The father is type O and mom is type A heterozygous. I mean, she's got one dominant, one recessive. So dad is type O, so he's 2 recessive, so 2 little i's. Mom is type A heterozygous, so she gets a big I and an A, and she gets a little I. So we're gonna do one, take my I's down first. I. Type my IA across. And I'm gonna put that as a superscript. And then take my little i's across. So parents' genotype, dad was II, little I. Mom was IA, little I. So the possible genotypes from this cross, well, we have right here is IA, little I. So that's one. So we get IA, little I. And we also get little I. So little I. Possible phenotypes, this one right here, IAI is type A. And then right here, little I is type O. Possible percentages, 50% is type A. And type O is 50%. So that is the rundown and the notes for complex inheritance patterns. So codominance, you have two dominant alleles, they both show up equally. Codominance, two dominant alleles, they blend together. And multiple alleles, you have three or more alleles that determine it. And the only one we're using for that is blood typing. So we'll do a couple practice problems today, maybe tomorrow, I haven't decided yet. And then these will show up on your assessment next week. So be prepared to do at least one blood typing problem on there. You do have an assignment for today in Canvas. So you should have gone through, we have the video right here, introducing codominance and incomplete dominance. Your notebook pages, we just went through all of those. So your worksheet here is, oh, it's time to go home. Okay, so for the first few here, you're going to tell me whether or not each description is codominant or incomplete dominant. So birds can be blue, white, or white with blue feathers. So I have blue, I have white, and then I have blue and white. So is that incomplete dominance or is that codominance? Flowers can be red, white, or pink. Incomplete dominance or codominance. So label all of those, okay? In smileys, eye shape can be starred, which is an S, circular, C, or circle with the star, which would be SC, okay? So write the genotypes for the pictures, phenotypes. Then you're gonna do a couple of Punnett squares. So show a cross between a star-eyed and a circle smiley. So use your genotype you wrote for the smiley here and the genotype you wrote for the smiley here. Work your Punnett square. All right, so the rest of these are smileys. There is one little bit of blood typing work here. So you've got 1, 2, 3 blood typing Punnett squares. And then we're going to go to incomplete dominance. So you have 1, 2, 3, 4 Punnett squares on incomplete dominance. All right. So codominance, you've got 1, 2, 3 codominance, 4 blood types, 3 blood types, 3 codominant, 3 blood types, 4 incomplete dominance. All right. So if you get into this, you find that you need help, please shoot me an email. We'll get you fixed up. Okay, guys, I don't want you to flounder through this. I want to help. This is fun. I love this. It's going to get even better because then we get to start looking at human inheritance and start getting ready for our genetic counseling assessment. So with that, I'm going to log off for tonight. Again, if you guys get into this, you have questions, please email me. All right. You're welcome to Zoom into class anytime. The Zoom link is up in the announcements. We'd love to see you. All right. So have a good day, and I will talk to you guys later.